correlation and causation in brain research, and our first talk of the session is by Ami Citri. Ami is an associate professor of neuroscience at the Hebrew University of Jerusalem, and his talk today is titled Correlations and Causal Relations in the Study of the Cellular and Molecular Basis of Drug Addiction. So let's welcome Ami.
Thanks. Okay. Thank you. And Ute, thank you enormously for the invitation to speak here. This has been such a treat up till now today. So when Ute invited me, then it got me thinking about correlations and causal relations in a bit of a more formal framework than I do in my daily life. I'm completely ignorant of philosophy, of history, so that it allows me a bit of freedom in saying things about these fields, which I'll do in very brief before I jump into possibly giving examples of how we approach this experimentally in my lab. And what I want to do, so in the first 5 to 10 minutes, what I want to do is propose that we should maybe think very carefully about the use of the word causality and consider maybe discarding it in the large part. And then, I'll try to amuse you with some stuff that we do. So when we think of causality, then I think that there really are a tiny number of examples in biology of true causality. And one kept coming up in the talks today, which is sickle cell anemia, where we have a very clear genotype and a clear phenotype and a very strong association between the two. And there are a handful of examples that are on par with that. But the rest of biology doesn't actually sit within this domain. And although it's very common to claim causality in biomedical research. And I'll give you an example of that, which it's fun to share this. This is an experiment that was performed last week in my lab. And we've identified a group of neurons in a brain region called the striatum and the substructure within the striatum that we have reason to believe could be involved in the development of OCD. So we set out to explore these neurons, and what we're doing here is activating these cells using what's called an optogenetic whatever. We're infecting these cells, expressing in them an optogenetic actuator, this light-sensitive channel that will activate these cells whenever we shine light on them. And then we can look at the phenotype of the mice. And what we're looking for is a phenotype of compulsive behavior. And in mice, this is often grooming. So mice do this sort of orofacial action, which is scored as grooming, and that's the mouse correspondent of OCD. So here you see the mice just doing their own stuff. These are 3 independent mice with, just hanging out with these light guides leading to the striatum. And then if you'll notice, then it turns from off to on, which it does right about now. And you see the mice changing their behavior completely, where you see that guy kind of rummaging around his mouth, and this guy moving between licking in the air on the floor and working with its hands and its mouth together. And then soon the light will be turned back off, and let's wait for that. All right, the light's off, and they immediately revert back to whatever it was they were thinking about before. So when we see this type of evidence, then it's compelling to think of causal statements, saying, okay, we activate these cells, and we see something that looks like an OCD-like behavior. Tick. We're good. We've uncovered a brain region because we had this prior expectation. Had I primed you with a different prior expectation, telling you, we think these cells make the mice nauseous. We think these cells make the mice want to expel some liquid from their mouth, or just, care about something else, some other explanation for what we're looking for, then you could have created the association in that direction, and introduced a different explanation for the observation. So that's just a subtle example, and I just used it as an excuse to bring this movie, which I think is pretty cool, for the misinterpretation of evidence as causality. And so one statement, and I'll make a few along the way, is that hypothesis testing does not equate causality, even though the two are often confused in the literature, because it's very easy to take our flow of thought from a hypothesis to some investigation that associates a phenomena with the hypothesis and therefore proof of causality. However, biomedical research populates a spectrum between these two binary definitions of correlation and causality, or if we can state this differently, all of biomedical research, almost, in the large part, 99.9% of biomedical research is correlational in its base, and it populates a large spectrum in terms of, now let's introduce a different metric, explanatory power. So we can, instead of defining phenomena as being either correlational or causal, I think we might want to think of them as being correlative in their nature. So the investigation that we perform is largely correlative, but then we can attribute to it some non-binary metric, some analog metric that in this, I'm suggesting the idea of using the terminology explanatory power in order to say how close we are to explaining what we're looking at, what we're investigating. And the explanatory power we can maybe break down into a set of criteria that is still qualitative, but perhaps allows us to differentiate a little bit more clearly what we're talking about. So better explanatory power could exist when a hypothesis that's based on correlation would account for many observations, would provide mechanistic insight, providing a higher accuracy of description, providing greater predictive power, making fewer assumptions, and is more falsifiable. So sort of cobbling together different philosophical musings, perhaps different philosophical approaches, somebody would have to tell me if that's the case, to populate this notion of explanatory power. So what I want to suggest is that maybe we should replace this holy grail that we're looking for in biology, which Ellen, in one of the breaks, very eloquently said it's asymptotic. We should aspire towards causality, but we'll never actually reach it. So maybe I think that maybe we should acknowledge that fact, and instead of aiming for causality, we should aim for explanatory power and populate it with these additional dimensions that allow us to explain what we've actually observed. Another example that I feel is relevant to rethink in word choice is necessary and sufficient, which also are often used with causality. They're apparently used in 3,500 papers annually. Use the terminology necessary and sufficient in order to explain the importance of their observations. And so this was brought up by these Yoshihara, and they go on a rant. I think it's a 20-page rant against using necessary and sufficient. They suggest that we should replace necessary with indispensable. I think that's a subtle shift, but sufficient definitely is an improper word, and they suggest replacing it with inducing, which I think is a much more precise definition of what we're looking at when we introduce a gene and want to make, well, normally what people do would make claims about sufficiency.
Necessary and sufficient belongs to mathematics.
I completely agree. Mathematics, physics, you can use this terminology within a framework.
Maybe.
Okay, but not in biology. And what I think maybe for the historians, maybe you can tell me if I'm going in the right direction. I think that all of this is based in the birthplace of contemporary biomedical research in the 1950s, which Ute was describing and came up in a few of the talks earlier today. So I think as biologists, we have guiding principles, whether we acknowledge them or not. And I'll discuss two of them. One is nothing in biology makes sense except in the light of evolution, which was stated by Dobzhansky. I think that's a crucial, fundamental pillar of how we approach biology, and it's constant. It stays solid. The other one, I think, was established in the 1950s, was a view of energetic efficiency being a key in biology, that biological systems are obsessed with being highly efficient in their utilization of the resources that they have. And I think this might be because, at this time the energetic cycles were being deciphered, the simple regulatory systems that Jacob and Monod described had a minimal set of components and could be explained in a very minimal set of moving parts, which made absolute perfect energetic use of what was available to it. And I think that with the genomic revolution and the identification of multiple copies of genes and of redundancies and the transition in the 1990s to mapping out circuit pathways and understanding that cells are cells, systems, organisms are obsessed with regulation to a much larger extent than they are with energetic efficiency, then we can also do away with the binary descriptors, which I associate. So energetic efficiency, if a system is absolutely efficient, then yes, you can expect possibly causality, you can expect necessity, you can expect sufficiency. But if a system is obsessed with trying to maintain homeostasis, if it will expend enormous energies and toss out, create proteins, destroy them, toss out a lot of energy just to maintain regulation, then everything becomes a little bit more nuanced in what you're looking for in a holy grail description of a discovery. And so I think that with the transition to looking at systems from a perspective of regulation comes this more nuanced view of understanding that we are looking at correlations and that perhaps we should change our terminology, again, going from causality, necessity, sufficiency, to maybe, something along the lines of explanatory power and the determinants that come with it. Okay? All right, just food for thought. Just some, since Ute's invitation, and I'm very, very happy to hear people's opinions. What's that?
I don't agree.
Yeah, that's great.
And not with your term of this Catholic term of Holy Grail. It was already discarded when it was used in terms of genomics. Please use another one.
Oh, okay. I'll think about that. Okay, so what I'd like to do with the time that I've left for myself for talking about science is amuse you with some stories from my lab that link a few other words in the description of the workshop. So to put it in perspective, what we're looking at is neuroplasticity. That's what my lab is interested in, which is in place in the brain to support adaptive behaviors. And we can think about this, an easy way to conceptualize this is when we think of babies that interact with the world, they interact with inanimate objects, they interact with their social structure to identify what is legitimate, what's not legitimate. They acquire habits that are adaptive, that allow them to maintain themselves healthy, or food preferences that, again, maintain them healthy. And I bring these two examples because these are two examples where you can easily think of how an adaptive mechanism can become maladaptive and form the basis for compulsive behaviors or disruptions in eating habits that are maladaptive and unhealthy. So we view it as being one substrate that underlies how we incorporate information from the world, forming our identity and defining how we will interact with the world in the future. Again, taking a page from tearing a page out from a philosophy book, then Schopenhauer said, man can do what he wills, but he cannot will what he wills, which I find quite cute. Okay, so our perspective is very straightforward, simplistic. We think of an experience as something that's represented here and now by neural activity within relevant brain structures, which then leads to expression of genes, which underlie a modification to neuronal function, which then underlie a modification to behavior. All of this assuming that the experience is salient enough, that it is defined by the nervous system as being important enough to be incorporated into a long-term change in the system. In my lab, we address two filters here. Two cognitive filters. The first is that of attention. So what do we attend to in the world now? What information do we want to allow to reach our deeper layers in our brain? And the second is memory. What do we want to take from our experience here and now and incorporate in a way that will modify our behavior in the future? And again, form the basis of our identity. And a large part of how we look at the incorporation of information in the brain to support experience-dependent plasticity is using this layer of information of gene expression. And what I want to share with you is one of the stories from my lab where we suggest that it's possible to decode the imprint of recent experiences using gene expression, or in other words, we propose that there's a layer of highly textured information encoding the nature of recent experiences in the brain, and this is found in inducible transcription. What got us here is our obsession with cocaine. We love cocaine in the lab. It's a very convenient experimental tool to use in mice, and it also has enormous social consequences. So, we at least have the potential to feel like we're doing something that may be important to somebody at some point. When we give cocaine to mice, then you see a very stereotypic behavior. So these are mice following daily exposures to saline. So you see, and we're testing how much they run around in an environment. So they become bored with these exposures to saline. They run around less and less, bored with the environment. And then one exposure to cocaine, acute exposure, you see this elevation of locomotion, and then daily exposures to cocaine cause an increased extent of how much they'll run around. After 5 repetitions, we call this chronic exposure. Then we leave the mice in their home cage with no cocaine exposure for a few weeks, bring them back and give them another acute exposure to cocaine. But now they've had past experience with the drug, and you can see that the response is elevated to the response that they created following chronic exposure, and perhaps even a little bit more, which is termed incubation of cocaine craving. So this is a very convenient system. You see here an associative memory that was formed between the drug and the context. This doesn't happen if you give the drug outside of the context. Yeah.
What is on the y-axis? The speed when you stay around?
The y-axis is how much they run around.
Does it mean that they sleep less, or
No. This is within 15 minutes.
within 15 minutes of them running?
Yeah.
And when do they sleep, or do they sleep more or less?
So this is one daily exposure. So we haven't measured sleep, but I don't think that it should be disrupted.
What do we have on the y-axis? These are weeks or hours?
No. These are days.
Days.
Daily exposures.
What's actually the activity pattern between day 6 and 25? Do they, is
Does it remain elevated without cocaine?
So this is transient. This elevation locomotion is something that is peaks within about 5 minutes, stays maintained for an hour, and then drops down to baseline within 2 hours. So when they're back in their home cage, then they're pretty much as they would be otherwise. So what we do is then we profile transcription within a number of brain regions of the reward circuitry of mice. This is a sagittal section through a brain of a mouse. And we're taking those brain structures that are very tightly associated with the reward circuitry, and we can dissect them very reliably. So we know that we can get a consistent tissue each time we perform the surgery. And this is what data typically looks like. So here we're looking at a couple of dozen genes on the y-axis, and then on the x-axis we have time following acute exposure to cocaine, chronic or challenge exposure, and this is 0 hour, 1 hour, 2 hours, 4 hours, and each column is an individual mouse. So the mice are chunked together by the experience that they had. And so I think this picture tells a very compelling story quite immediately. One is that, not a whole lot happens throughout most of the brain, but you do see gene transcription within dorsal striatum, nucleus accumbens, these central regions of the reward circuitry. And then when you look within the 1 hour time point in either one of these, and I don't know if you can see it, but these are a bunch of columns clustered together, so a bunch of individual mice. And when you consider that these are all individual mice with different life histories, and they share one experience recently, then you can see that their gene transcription is extremely robust. And this is actually the most robust parameter that I've witnessed in neuroscience. In neuroscience, we're used to a lot of individual variants and variance in responses, and here you see something that's very tight. And it suggested to us that this might serve as a readout we can use to look at the encoding of experience in the brain. So we set out to test that, and this doesn't look that good. So we looked at a couple of genes. This is comparing acute exposure to cocaine to this acute exposure in mice that had prior experience, what we term challenge. And we're looking at this immediate early gene FOS, and you see that it looks identical in this structure that we're looking at. These are the individual mice. But then there's a different gene, this immediate early gene EGR2, that shares the same dynamics between the two, but completely distinguishes between mice that were exposed the first time to cocaine from mice that had prior experience with the drug. So this suggested that maybe we can, if we get enough information, we can open this up and look at the encoding of recent experience. So we used another very simple paradigm, which is reinstatement of feeding. You take the food away from the mouse and give it back to them, and then look at transcription following a time course. And again, this gene FOS cannot differentiate between these three experiences. The second gene, EGR2, cannot differentiate between the acute cocaine and feeding, but now a third gene can distinguish between these three situations of cocaine experience and feeding. And so 2 years later, the work of a bunch of people in the lab opened up this investigation to a variety of different experiences, ranging from very low salient, so this is saline exposure to a novel environment, a foot shock in a novel environment, an aversive experience of lithium chloride, repeated lithium chloride, cocaine experiences, exposure to sugar water, re-exposure to sugar water, and this feeding. And here we're looking at gene expression in these polygons that expand in log scale, log 10, where each axis corresponds to a different gene. So it's a minimal representation of this gene expression that provides a landscape where you can immediately see the aversive experiences having a larger representation in these structures, the rewarding experiences having a more prominent representation in these frontal structures and limbic structures that are involved in reward. So we get the impression that we can segregate recent experiences based on a subset of gene expression across structures.
So do I understand correctly that to form these quadrilaterals, there are 4 genes, right?
Yeah. 4 genes, and their expansion is in log scale corresponding to the level of induction.
And if it is, imagine that something is downregulated, what do you do then?
It isn't, but it would collapse, yeah, because it's in log scale, then it would collapse. So we profile many genes, but we chose these 4 genes that we can represent in this way just to give this one unified picture.
But we have genes that are transient. What do you actually plot? The maximum length?
This is the amplitude at 1 hour following the experience.
At one specific time.
Yeah,
Could you say
which
what
is the maximum.
Could you say what the 4th gene is? It's really hard to read.
So it's all published, but this is EGR4, EGR2, FOS, and ARC at the bottom. Yeah. When we look at this a little bit more in detail, then we can see things that look like representation of negative valence. So if we're comparing here between, we introduce the mice to this environment that's pretty intriguing for them, which has a grid on the floor, and so we see its representation across these brain structures in gene expression. Then when we introduce a foot chalk in this environment, then everything looks the same except for this induction in the amygdala, which is famously involved in processing of aversive experiences. So that makes sense. Then when we look at this aversive experience of lithium chloride, then we see this representation across structures. But if we repeat this, so mice are exposed repeatedly to this aversive experience, then you see a decrease in its representation across these structures, which might correspond to habituation to a negative experience that's unavoidable, kind of like elections in Israel that we become desensitized to if they occur frequently enough. And then the opposite you get when you look at cocaine experience. So you see an expansion of the representation in these 2 structures, so the frontal cortex and dorsal striatum. I won't bore you with our interpretation of that, but when we overlay the 2, then this corresponds to some interesting associations that have been made with the role of prefrontal cortex, where it could be involved in the initial evaluation of whether an experience is salient, whether it's important, and then the representation of the lithium experience is diminished while the representation of the cocaine experience is expanded. And the opposite is true for the nucleus accumbens, which is involved in implicated in reward prediction, where initially these experiences are novel, but once they're expected, then the representation becomes very similar. So the expectation is reinforced, and that's what's known in literature, and we see some signature that could correspond to that in the gene expression. So, you know, flying a bit further with the interpretation, then this is kind of what we're hoping that we're seeing here, is that the representation here, let's say, of repeated cocaine could correspond to the mouse putting together a sentence for itself saying, wow, just like I remembered, I want to experience that again, corresponding to the induction throughout the different structures, and the opposite with this foot chalk. So this is an environment, a weird, novel, interesting environment where I have an aversive experience of foot chalk, definitely worth remembering. So we view this transcription as being always prospective, as encoding or representing the experience now with the valuation of what its impact should be for future behavior. When we feed this data into a machine learning algorithm, then we get 94% efficiency in decoding the recent experiences of individual mice, so we only get some small misinterpretation of the algorithm confusing the sucrose experiences, but besides that, this minimal data set is sufficient in order to decode the recent experiences. And when we look to see whether the information that's important is embedded in structures, so whether the data from each structure alone is sufficient for the decoding, we see that it is not. Or whether it's within genes, so we see again that of the 5 genes we chose across the 5 structures we chose, the genes by themselves are also not sufficient, but a couple of them are not terrible. So EGR2 provides 70% accuracy, and FOS also around 70% accuracy. So to summarize this part, what we see is that salient experiences are encoded by unique transcription patterns in relevant brain regions, and we propose this concept of behavioral transcriptomics, which has a number of benefits in investigating the encoding of experiences in the brain. And our vision is, and I don't know if you guys care for this too much, but it's sort of for the way we're looking at this, is that the nuanced features of an experience, so perhaps we can segregate experiences across axes of salience, valence, novelty, or expectation, or possibly other axes, are encoded by the expression of a subset of genes in relevant brain structures, and that these subpatterns of transcription may be driven by subsets of synaptic inputs or features of the network, and then could feed back and impact these elements that induced them. So we would have a correlation between how the network organization impacts transcription and how the inducible transcription would be responsible for modifying the network in response. So I'll take the few minutes that I have left to describe our attempts to go further towards explanatory power. This used to say causality, but I listen to myself sometimes, and I replaced that. So we're asking about the spatial distribution of these genes. So up till now, this was hunks of tissue, but we want to know what cell types they're in, how well they're correlated with each other, and is their expression important? Is it relevant? So for this, we use SM-FISH, which is a technique that I'm sure Shalev Itzkovich will describe tomorrow. It's single molecule fluorescence in situ hybridization, where we see the co-induction of these immediate early genes within the same cells following cocaine. And we see that they cluster in 2 regions in the stratum, which are this medial region and a lateral region, the lateral region has been under-investigated, so it was intriguing for us. Within the stratum, there are 2 main types of neurons, direct pathway and indirect pathway. Luckily, a very simple logic to the system. One, the direct pathway pushing go, so motivating behavior, driving motor action. The indirect pathway contrasting that. And what we observe is that in this ventrolateral region, expression of this EGR2 gene is induced selectively within D1 neurons. Don't bother trying to decipher this, just take my word for it, and I can discuss it later. And then, so we wanted to address the activity of EGR2 within ventrolateral striatal D1 neurons to see whether it plays a causal role or plays I caught myself there, whether it has a function in promoting the cocaine context association, so a slightly more advanced version of asking whether a mouse cares about the environment in which it received cocaine. And for that, we inject a virus into this region, expressing a dominant negative version of this gene EGR2, and we see that the mice develop a lower preference for the cocaine-coupled environment, suggesting that maybe this gene plays a role, this gene within these cells, within this structure, plays a role in encoding context association of the drug. And then we have a bunch of other supporting data, which I did not want to bore you with, but it provides explanatory power in support of the hypothesis that EGR2 expression in this region is important for development of cocaine preference. So I shared with you our notion of behavioral transcriptomics and our attempts at a functional investigation to look at the role of EGR2 within these D1 neurons. And I'll summarize by just going over what we've done. So we started off with omics type data that provided the basis for our hypothesis that recent experiences are encoded in patterns of IG expression across relevant brain structures. Then we replicated this across multiple biological conditions, and this provided further support for the hypothesis. Then we performed perturbations of EGR2 and the EGR2 expressing neurons, which I didn't share with you, that led to the conclusion that these neurons and their expression of EGR2 are important for the development of cocaine preference. Yeah, and that's where what I would like to term now our explanatory power currently ends at this correlation between the induction of EGR2 in these cells and this perturbation of EGR2 that reduces the context association of cocaine. So these are the heroes of this story. This is the team in my lab that works on striatal structures and behavioral transcriptomics, which was the project of Diptendu Mukherjee, who's now finishing his PhD, and Bogna, a postdoc, who left the lab. Ben and Diptendu are further doing the work to look deeper into the function of the genes within the ventrolateral striatum. And when Bogna left, then David joined, and he's the one who's working with Ben on the OCD-type behaviors and potential role for these neurons in there. And the second part of my lab works on this brain region called the claustrum and its role in supporting selective attention and reward. And just to show you that Jerusalem has a beach, and we're very proud to know how to swim, which is what Jerusalemites are always kidded for not knowing how to swim. Then nobody drowned. We all made it back to the lab.
That's actually Bersheva's beach.
You're right. You're absolutely right. It is Beersheba Beach. Yeah, and this is our funding. Thanks.
Okay, so we have 8 minutes for Q&A.
I don't mean to monopolize all the discussion, but when you do these perturbation experiments, which I would have loved to hear more about in detail, the question is how many of the other effects on the expression of the other genes can you monitor? That is, you're doing a perturbation and you're monitoring behavior, but do you know whether these effects involve only changes in EGR2 expression, or is it possible the entire circuit is collapsing?
Yeah. So we haven't done as much as we should on that, but what we do see is that when we manipulate EGR2 levels, we do see changes in all the other EGR2, all the other IGs as a consequence of that. Yeah, it's not simple.
It's starting.
Yeah.
You mentioned as one of the biological principles, Dobzhansky's dictum that nothing makes sense except in the light of evolution. I mean, I personally don't agree with that, because there is a lot of things where you don't need evolution in order to understand certain mechanisms, etc. But this is not the point. I wonder how it applies to your own research. Where does evolution come in? Or do you violate that principle?
So first of all, we use mice as we treat them as little people, and so we're just making the extrapolation from mice to humans. So I can't say that that I interpret my observations as potentially having implications for humans because I assume that No, yeah, I'm just hand-waving. I don't have a good explanation for how we use evolutionary thought to influence our approach to neuroscience.
Okay, there are other things we can perhaps discuss later. I don't think anybody claims that any hypothesis leads to causality. That would be really stupid. So,
That's what I'm afraid that people do. I mean,
I don't. But
I was just looking this up. Well, I was aware of this in the past couple of weeks, and I noticed how many papers have a causal role in their title, coming out in
right, but not just because they have an hypothesis. I mean, it's, let's say I argue in a very narrow way. But we can maybe discuss that
Okay.
later.
No, I agree with you that this shouldn't be the case, but I sadly think that it is the case that people allow themselves a lot of flexibility in their terminology.
Thank you for your very interesting speech. Are there any models about behavioral addiction versus chemical addiction, substance addiction? Are there any differential models?
So, behavioral addiction, you mean like internet addiction, sex addiction, all these...
Sex addiction about mice?
Yeah, so there aren't very good mouse models for gambling addiction or sex addiction. People in the human literature, then people, I think, largely want to make the claim that the two, that there are equivalences between drug addiction and psychological addictions, but yeah, the data isn't as solid as, you know, the mechanistic evidence isn't there as it is for drug addiction in rodent models.
There's time for one more question.
I mean, thank you. That was a lovely talk. Your data on the sensitization of the locomotion to the cocaine administration suggested that that was lasting over a period of weeks,
Right.
if I understood correctly. So is the implication then that these patterns of gene expression are also persisting over that period of time?
So let me, I probably didn't explain that part well enough. So the changes in locomotion are context dependent. So it's the association of the drug of abuse in this particular context which acutely induces an increase in locomotion. And so if you give cocaine repeatedly and then re-expose in a different environment, you won't see this. If you expose a mouse that had experience of cocaine in this environment now to saline, you will see the elevated locomotion. The changes in transcription evolve with the exposures. So we see a different pattern of transcription following chronic exposure than we do following the challenge exposure. And we haven't measured very carefully during this period of abstinence between the chronic and the challenge, so I don't know to tell you how it evolves within there, but it does. So there's some reorganization of the circuitry, and this has been also measured using electrophysiological parameters to see that there's a shift in how the circuit organizes itself between the chronic exposure and during this period of abstinence from exposure to the drug until the mouse is re-exposed. I hope that answers.
Well, so is the implication still that the persistence of the association with the context implies some memory that has been created over that period of time? Is that?
So the memory, basically it's sufficient to expose a mouse once to a context, then remove it from that context and come back 2 months later and it will have some context association with the drug. Mm-hmm. And yeah, the assumption is that this is a memory of this contextual association that's stable over time. But you don't necessarily need the repetitions in order to create that memory.
But the memory is associated with the expression of these genes? That's the implication here?
So the Okay. So the expression of the genes is they're induced acutely in order from our perspective to encode the experience that happened now, and then they terminate. If you will re-expose to this environment, you get a re-induction of genes, but then it depends on where you are in the time course following the initial exposure, how you re-expose them. But it's always prospective. So it's not a maintenance, but there's...
Some memory.
Yeah.
Thank you.
Unfortunately, we have no more time. Let's thank our speaker.